Okay, second. Okay, hi everyone. Happy Saturday, wherever you are right now. Maybe it's Sunday somewhere. Uh, well, welcome to our first official journal club event of this year. I'm not sure if anybody had attended our special event with the program chairs a couple of weeks ago, but that was a special event. So this is our official journal club. And to start off this year, we have a very special guest today, Dr. Mengjin Dong. Dr. Dong finished her PhD in August 2023 at the University of Pennsylvania, where she was supervised by Professor Paul Yuskevich and David Woke. And she is currently a postdoctorate fellow at the University of Texas Science Center in San Antonio, where she's working with Dr. Mohammed Habis. Dr. Dong's research primarily focuses on developing robust deep learning methods for longitudinal image analysis of Alzheimer's disease progression. She is interested in using modern machine learning based techniques to understand the mechanism and interaction of different biomarkers within Alzheimer's disease and related dementias. In addition to her dissertation work, Dr. Dong has also spent time as research intern in Siemens at Princeton and also at Amazon in Seattle. Well, with that, I would like to welcome Mengjin, and Mengjin, you can take it away. And also, if you have any questions, you can directly put them in the chat box.
Yeah, thank you. Thank you for the nice introduction, and thank you for inviting. I'm very happy to participate and share my recent work to this group. So today I'll be talking about mainly like my work with Alzheimer's disease progression that was happening in my PhD, during my PhD and postdoc time. And the topic is detection and localization of progressive changes in longitudinal MRI of the hippocampal region in Alzheimer's disease with deep learning. Just to make sure people can see my screen, right?
Yes.
Okay, thank you. Yeah. So Alzheimer's disease is one of the fastest growing threat to the public health. As estimated by NIH, every 65 seconds when this has passed, someone in the United States will be diagnosed with Alzheimer's disease. And the number of AD cases will grow tremendously from 5.7 million currently to maybe 14 million by the mid of the century. Currently, a lot of clinical trials are being developed for Alzheimer's disease treatment, and we can see there are plenty of phase 1, 2, and 3 clinical trials that are landing in different perspectives to treat Alzheimer's disease. And just want to give some introduction background of Alzheimer's disease. The hallmarks of Alzheimer's disease will include intracellular neuronal fibrillary tangles and also amyloid plaques. So at the very beginning, neurons are healthy and in the brain, but then later, due to ineffective clearance of amyloid plaques, that is a toxic protein that's happening in the brain during metabolism, the amyloid started to deposit in the brain, and it will be bad to the brain. And then this will lead to cell death, like neuronal cell death, which will lead to brain structural shrinkage in the brain. And then later, when the shrinkage accumulates, that is where memory loss or other effects that can happen in the that can be detected by the human. That is like when we can actually find out that, oh, this person might have Alzheimer's disease. And one thing about Alzheimer's disease research is that currently Alzheimer's disease progression cannot be resolved yet, but we have already developed drugs to slow down the process of Alzheimer's disease. And one of the challenges for developing such treatment for AD is that we don't have reliable in vivo biomarkers to monitor the slow disease progression, which is maybe more than a decade before we can find clinical like symptoms related to AD, especially in the preclinical AD stage. So for the denomina drug that has developed, we found that the earlier we intervene, we conduct intervention in this for patients, then the better effect we will be able to achieve for delaying the disease progression. So that is why we want to target people in early stage as possible. And then we also evaluated a couple biomarkers that are associated with Alzheimer's disease to find the good intervention stage. In this plot, we have, I guess, people who are working on Alzheimer's disease research will be familiar with this plot, but maybe not familiar for some other audiences. So I just give a brief introduction. So this plot shows the difference in clinical disease stage and when we can discover these changes from a biomarker perspective. So at the very beginning, A-beta protein will deposit in the brain, and this can be discovered by PET imaging. However, PET imaging is low resolution and is also very costly and is not covered by US insurance yet. And then later there's tau-mediated neuronal injury, and similarly, we cannot detect, they are very blurry for progress tracking, and the cost is very heavy. And then later we have our brain structural change and the memory and clinical functional change. The ideal time range to detect these changes is in the preclinical AD stage, where it's highlighted in this like light red square, and that is the earliest detectable stage of Alzheimer's disease. And studies have also found that with temporal structural MRI measurements such as temporal composites and hippocampus volume, we will be able to receive the lowest sample size for clinical trial detection. So that's why we want to, in this study, we're focusing on hippocampus and a medial temporal lobe area in this study. And when we're trying to measure the hippocampus growth progression, we found that hippocampus and the medial temporal lobe, which is located here in the brain, is the earliest detectable structural change in patients with Alzheimer's disease, and then gradually it will expand to the whole brain and other areas. And that's also the earliest deposit of amyloid and tau proteins in the brain. In past studies, we have found that although both in normal aging, normal elderly, and in advanced Alzheimer's disease, the hippocampus volume will have a shrinkage effect. The shrinkage rate for these two groups are significantly different. And also in while the one of the counterintuitive thing that was happened is that for the denomina clinical trial, we found that for placebo group and the clinical trial group, the shrinkage in the hippocampus volume are the same for people with drug and without drug. So it's also worth investigating. And so that's why in this study, I am trying to use hippocampus volume measurements to track longitudinal progression of Alzheimer's disease. And when we are measuring longitudinal progression, there are in general two ways to measure it. The first way is shown on the left. This is like when measuring kids' height. We can directly treat each measurement independently and directly measure their height, and then calculate the difference by subtraction. And then the other way is to measure it longitudinally, where we directly measure the difference. And the benefit of this is that we are minimizing the error that can be measured in this process. And then in previous studies, we have looked at longitudinal measure, cross-sectional measure versus longitudinal measurement for the hippocampus volume at different disease stages. So the green box is normal control, and then the more red it goes, and then the heavier the disease will be. And then we found that in clinical, in cross-sectional studies, we didn't find any difference between the control group versus preclinical AD group or other subgroups of preclinical AD. While if we conduct this analysis in a longitudinal way, we were able to find a difference between the normal control and the preclinical AD group, and as well as the tau positive preclinical AD group, which is a subtype of preclinical AD. And from this, we can see that longitudinal measurement is more sensitive to group difference compared to cross-sectional ones. And in the past, the way to measure longitudinal progression is by deformation-based morphometry method. This is an algorithm that was developed in my PhD lab at Pixel at UPenn, where we are measuring like the change directly in a longitudinal way. So when we have the baseline image and the follow-up image, we perform an image registration where we conduct a rigid registration plus a deformable registration and obtain the deformation field. And then we also perform baseline image segmentation. This was done by an automatic image segmentation tool called ASHES. And then we will apply the hippocampus mask and integrate it to the deformation field. And then we will be able to summarize an atrophy rate for this image pair. And then the benefit of this is that the algorithm carefully deal with numerical calculation, because this is large deformation. This is a large like deformation field, so it has a lot of numerical errors in it. And also this is unbiased in the way that the rigid registration happens in the halfway space rather than in the baseline space or the follow-up space. However, the challenge of this measurement is that artifacts in individual time points can introduce noise in atrophy measurement. As you can see, this is a T1 weighted MRI image, and the hippocampus is roughly in this area. And we can see that the images get blurry in vivo, and there's no ground truth, meaning that we don't know exactly where the hippocampus volume can be. And the result is that algorithms are unable to distinguish biological changes that we want to measure and image artifacts. Actually, as the result of the ALOHA measurement that I have described, although that the hippocampus volume is expected to shrink monotonically over time, which is expected for elderly people, but not adults, young adults currently. The ALOHA measurements actually measure roughly a quarter of positive hippocampus hippocampus expansion areas, and this should belong to some measurement difference. And in another study where the researchers conduct analysis for 5 different algorithms, and then they also found that the percentage of hippocampus growth will vary from 20% to like 40%. And also the detected subjects for hippocampus growth are not overlapping a lot for these different types. So which means that it's highly likely that these subjects, these measurements will be due to measurement errors rather than biological changes. So our hypothesis for our study is that the measured change from MRI can be decomposed to two parts. The first is biological change, is what was really happening in the brain, and this is what we want to measure. And the second part is non-biological change that might be due to motion, imaging artifacts, head position, or scanner differences, etc. And we observed that the difference between these two might be that the biological change can be dependent of time, while non-biological change might be random and they're independent of time. And our hypothesis is that we want to use deep learning tools to detect biological changes in the green box and ignore non-biological changes that are in the red box. So here I will come to the first project that I developed to detect subtle changes in longitudinal MRI in AD. So we're trying to observe some features of biological change in the brain. And the first property is the directionality property. That is, like, in the context of aging, hippocampus volume is only expected to shrink monotonically a long time. And the second property is the magnitude property, which is the hippocampus volume loss is expected to be approximately proportional to interscan interval for individual subjects. So in this formula, the change that are happening between two images at different time points, A, B, and C, should be almost proportional to the time that has elapsed from those subject scan times. And the idea of the deep learning model is to use time-related information that is described in the directionality property and the magnitude property as a surrogate to neurodegeneration with deep learning. Yeah. So to solve this issue, we designed those properties into two loss functions. The first loss function is the scan temporal order loss. This is very straightforward. So if we want the deep learning model to tell the directionality of two images, we will design a loss in which we will arbitrarily orderly input two images. So the baseline image could be the first one and follow up the second, or it can be reverse order. And then we let the model to tell which image is scanned first and which image is scanned the second. However, the algorithm was able to detect this property really well, and the accuracy can go up to like more than 90%. However, the problem is that the algorithm makes no difference for like however long the interscan interval is. So the confidence interval for the algorithm can reach the same high whether the scan is 3 months apart or 3 years apart. So this is not useful for measuring progression. This is where we try to come up with a second idea with the magnitude property. And we make the model to answer the question of which scan pairs have a longer interscan interval. For this, we replicate the first network here, and then we also arbitrarily orderly input additional images from the same subject. And then the model, we designed a relative interscan interval prediction. And with this, we will be able to predict the change between two images. And we use the time difference as the ground truth for the model to predict. So the idea is that with this RISE loss, relative interscan interval loss, we will predict the change that are happening between these images, and then this can be the ground truth that can lead to the model training and can guide the model training. After this, we will have some accuracy for both losses, and our ultimate goal is to find a measurement to any question? Yeah, so it's to find a measurement for how severe the disease will be. And our motivation is for the measurement to be proportional to disease progression, similar to atrophy rate in ALOHA measurement, and so that it can be served as a surrogate biomarker for disease progression. So suppose that we have a normal control patient and Alzheimer's disease patient, and both of them are scanned 500 years apart, and this is the actual scan time interval between an image pair, and we want the model to predict a scan time interval that is very similar to real ones for cognitively normal patients, and then to predict accelerated brain atrophy, which can be represented by a longer scan time interval predicted by the model with Alzheimer's disease patients. And the way we achieve this is that we perform a linear regression model to this analysis. So we took the last layer of the activation layer as factors here, x1 through xn, and then the y-axis or the predicted variable will be the actual time that are spent in this interscan. So with this, we will predict an interscan interval for each image pair. And then the rate of progression will be represented by the predicted to actual interscan interval ratio, which is the predicted interscan interval divided by the actual time elapsed. And just to mimic this plot, the pair for the cognitively normal patient will be 0.98, which is close to 1. And for AD patient, it'll be 1.4, and it's slightly more greater than 1. Yeah. For this study, we use T1 weighted MRI of the hippocampus from ADNI2 and ADNI-GO patients. We split the training set, we split it into a training set of 155 subjects and test set of 336 subjects. The reason that there are more images in the test set is that we want to perform a group-wise analysis of progression rates for different disease stages to see if we have a significant difference or not. And with that, we need a distribution that will require like a large number of samples. And we also put all the preclinical AD images to the test set for the same purpose. Our results show that for the two losses that we are conducting, DeepAtrophy has a higher scan temporal order accuracy compared to traditional deformation-based algorithm ALOHA for all disease stages. And we also observe that as disease gets more severe, the accuracy goes up. I think that is explainable by like when the disease gets more severe, there are more changes in the brain, and then the noise can be like more, can take part of a less ratio compared to the biological change. That is why the accuracy goes up. In comparison, in other algorithms, there are like 20% to 40% accuracy. Our algorithm retrieve only like 10% error. So this is significant advancement compared to other algorithms. And then when we look at the second loss accuracy, we found similar results that DeepAtrophy has a higher accuracy, and also the high accuracy goes up as the disease gets more severe. We also plotted a spaghetti plot for ALOHA DeepAtrophy with only the first STO loss and DeepAtrophy PII prediction, predicted interscan interval prediction. We found that with ALOHA volume difference is, yeah. Any question? Yeah, I guess I'll continue. So with ALOHA volume, the lines are more straight because they are more like numerical analysis, but we found that with deep learning models, the lines are more like maybe sublinear because of the ability to detect like differences. But the accuracy rate for deep atrophy actually decreased a lot from ALOHA measurements. And then we performed the group-wise analysis difference study, and we found that for different stages of Alzheimer's disease, when we look at age-adjusted deep atrophy pair, we found that only deep atrophy can detect a significant difference between the normal control group versus the preclinical AD group, while ALOHA measurements and another neuropsychological measurement, PEC, cannot detect a change rate, indicating that deep atrophy model is more sensitive. And also notice that this is a study conducted within 180 days to 400 days, which is within 1 year. So this study is really effective. When we look at within 2-year studies, the same result hold, and deep atrophy has a much lower p-value compared to the other 2 groups as well. Now we look at the sample size required for deep atrophy and ALOHA. We found that for a hypothetical 1-year trial and hypothetical 2-year trials, in general, deep atrophy will have a lower sample size compared to ALOHA, meaning that we may need to recruit less people for a clinical trial if we use deep atrophy as a measurement compared to previous other measurements. And to summarize, the takeaways from deep atrophy is that it has higher scan temporal order accuracy and interscan interval prediction accuracy, and it appears to be distinguishing true biological change from noise. This result could be a more sensitive disease progression biomarker for clinical trials. However, it predicts only a progression score with deep learning backbones, which lacks interpretability. I'm going to pause here to see if anyone has any question or not. If not, then I will continue with, does Alvin has a question?
Yeah, sure.
Yeah.
Thanks for the presentation so far. I think, could you explain a little bit more about the prediction of the interscan interval and how you use that to measure severity of disease? I didn't quite get it.
You mean
the next slide?
this slide, this one?
Yeah.
This
Yeah.
one.
The previous one, how you measure like severity of disease based on the predictions.
Yes, yes. Yeah, so this one is just a plot of what I hope for in this measurement. And this is an like an illustration plot that we want so the idea for this model is that we want the model does not know. The model only knows that 2 scan pairs are input to it, and it does not know any information about the disease status. So some people will have accelerated atrophy, and some other people will have normal progression. And then the model would assume everybody have the same rate of progression-ish. And then what the model can do is the model can predict how long the time has elapsed from those scan pairs. And our assumption is that if one person have a fast progression, and then the time that has elapsed from the model will be longer.
Yeah.
And yeah, here I forgot to emphasize this detail that this linear regression fit is only fit on the cognitively normal groups. So yeah, so these are only for the cognitively normal people, but for cognitively like impaired people, this predicted like time difference will be longer, and that is why the numbers are larger here.
Oh, okay, yeah, I see.
Yeah.
Makes a lot of sense. Thanks.
Yeah. Thanks for this question. I think it clarifies a lot. Yeah.
And just like to confirm, it's like
Yeah.
you train this model that predicts the interscan interval entirely on cognitively normal people. You don't train it on data from sick patients.
No, the training is so the idea is that we don't know the progression rate at all, and we don't assume we don't let the algorithm to assume a uniform progression rate for different individuals. And that's why, like here, we only let the algorithm to predict the ratio between two changes, rather than predict the exact amount of difference between two scans.
Okay.
And that is we don't let the algorithm assume anything with regard to progression rate at all. So in the training process, we actually trained the model with all disease groups besides the preclinical AD group, as explained like in this here. But when we are calculating the for this calculation here, we only use on the cognitively normal group, but not other. Yeah.
Oh, okay. Yeah, got it. Thank you.
Yeah. Okay. Yeah. So the motivation for the next project is that we want to develop an algorithm that can link deep atrophy progression rate prediction with regions in the images to make sure that the deep learning prediction actually makes sense. So here is this is also a motivation. Rather than predicting only one score or one number for each image pair, we would like to predict something that's informative to regional information. The one intuitive approach is that we synthesize noise-free images. So when we are inputting arbitrarily ordered images, before inputting these images to this registration model, we synthesize noise-free images in between, and then the output will still be the medial temporal lobe volume change. And here we can plug in scan temporal orders or relative interscan intervals to this synthesized noise-free image network to guide this prediction. And then these images will update MTL volume estimation. Our preliminary results show that while the model still can predict significant intensity difference in regions with higher structural change, and they have neurodegenerative meanings in downstreaming analysis, we found that synthesized images are not realistic, and we don't know a very high standard of ground truth, and we cannot define noise-free images. So we abandoned this algorithm, and we look at the previous algorithm again, and we found that so in the past, we were using anatomical regions for image segmentation, but this is based on our human experience as well. So why not that we just change this part and let the model to predict regions in the brain in this hippocampus area by itself, rather than using manually delineated ones. So here is the original deep atrophy model. In that we still use a two-branch approach where we have the baseline and follow-up image that are input to the network in arbitrary order. And then we use Aloha model to generate a deformation field. And also now we relax the manual segmentation part to an attention network, which is a 3D UNet, and then the network will be able to predict shrinking areas and expansion areas at the same time. And with that, we use the same strategy to integrate network predicted areas with deformation field to get the total volume change. And this scan temporal order loss that we developed in the past will be back propagated to this attention network to guide this attention map. So this is the regional deep atrophy algorithm with scan temporal order only. And the key idea for this algorithm is that we use deformation-based morphometry method to obtain deformation field here, and then we use temporal-related information, which is the scan temporal order loss, to guide attention map generation. And then for the whole RDA process, I believe that you can already guess it. We just add this VC loss in between two subnetworks, and then we derive this magnitude property of which scan pair has a longer interscan interval. And with this, we are calculating delta V, which is the total volume change for both the shrinking area and the expanding area to guide the prediction for these areas. And in the test stage, we don't use, we only need to use one subnetwork to predict total volume change and obtain annualized atrophy rate from an image pair. So we don't need to use duplicated networks. And we conducted experiment using the same data set that we used before, and we compare results for Aloha and deep atrophy, which is the model in project 1. And for this project, for the second project, we add RDA with STO only, like the first loss, and the full RDA model. And we found that RDA, which is regional deep atrophy, keep the same as scan temporal accuracy and group differentiation ability. So in this plot, we calculated the AUC of two groups, and we found that with RDA, it has the highest AUC, and it's also more significant compared to other models for clinical sensitivity analysis. And then if we look at the volumetric changes, starting from the baseline, we have Aloha, deep atrophy, RDA, STO only, and RDA or. We found that the Aloha has straight lines, like we want to have straight lines because we're assuming linear progression for hippocampus. And then with RDA, we will be able to have a more linear relationship compared to deep atrophy. This can be observed maybe more clearly in this plot. Yeah. Lastly, we calculate the average heat map of RDA. So in the middle is the heat map on A- control groups. And then we also have a template and segmentation as a guide. On the left is the heat map difference of different disease stages of preclinical AD, early MCI, and late MCI compared to A- control group. And we can see that as disease progress more, the highlighted red areas, which is regions with more detectable shrinkage, are more significant in more severe diseases. And also for shrinkage areas, the same result hold. And we found that in the shrinkage area, the mainly area for us to find the difference is the hippocampus, as well as some peripheral white matter areas. While in expansion region, it's mainly in the gap or the C cerebral vascular, like the ventricle areas. The takeaway from The second project is that it automatically detects regions with consistent shrinkage and expansion in the medial temporal lobe area, and shrinkage regions are in MTL and peripheral white matter, and expansion regions are in CSF and ventricles. And we also found that RDA has better time inference accuracy and sensitivity to group differences compared to conventional longitudinal measurements. So this is the second project. And as a side project, I'll just, in case some people are interested, I'll just briefly go over it. And this is when I compare T1 and T2 weighted MRI image for detecting longitudinal atrophy for using only the DeepAtrophy model. If we look at the difference between T1 and T2 weighted image, so T1 weighted image is suitable for whole brain analysis, and it has more stable image quality, but it had lower image resolution. T2 weighted MRI has really high resolution on coronal plane, but low resolutions on the other two planes, and it's specific for hippocampus subfield analysis. And sometimes the scans are failed more frequently compared to T1 weighted image, so it has unstable image quality. By visualizing this, we really can't tell which image modality will be more suitable for our analysis. In the past, we found that T2 weighted MRI was shown to perform well in tracking out AD progression, but the analysis was conducted only on a small data set. As ETNI now collect high-resolution T2 weighted MRI images, we want to conduct this comparison for T1 and T2 weighted. So here are some details when we are trying to conduct T1 and T2 weighted comparison. They are specific to ETNI data set, so I am not going through it too much. If you want to know more about it, we can communicate afterwards. And this is also specific to ETNI. I'll jump directly to the results. So here, this is the characteristics of T2 weighted MRI. With this, I actually use less subjects for training and more subjects for test. And we found that for STO accuracy, although DeepAtrophy is always better than Aloha, we found almost no difference between T1 and T2 weighted image. So for T2, it's 88% accuracy. For T1, it's 89%, so almost indistinguishable. But for RISE accuracy, the second accuracy, T1 performs significantly better than T2. And both of them are significantly better than Aloha prediction. If we look at group difference for 1 year, we will find that T1 and T2 have roughly the same group difference. However, DeepAtrophy performs better than Aloha for both T1 and T2 weighted images. The takeaways for project 3 is that overall, we think that T1 is slightly better than T2, and DeepAtrophy performs better than Aloha. So the factors to contribute to this result is that the pro for T2 is that it has good image quality. However, the shortcoming for T2 is that probably the upsampling strategy in our analysis can be improved. And also we have less training data for T2. I think this result show that high resolution T2 has a great progression estimation, and it has potential to be a better longitudinal biomarker, but we still can't tell any difference. I mean, not too much difference between T1 and T2 weighted images, partly because that T1 and T2 are not collected at the same time for ETNI data. So we can't make a strict comparison. To summarize, I developed novel approaches to longitudinal MRI disease progression measurements by taking out only true atrophy and ignoring noise. And deep atrophy can be served as a surrogate biomarker, and it's very sensitive to preclinical AD stage. Regional DeepAtrophy can characterize regional information of AI decision progress, and both T1 and T2 weighted MRI can be suited for longitudinal atrophy analysis. And we hope that these novel loss functions can be applied to other longitudinal progressions, such as tracking multiple sclerosis lesions. Some of the future work will include combine multi-image modalities of T1 and T2 to do analysis, or synthesize noise-free images with scan model or diffusion models in the future. If we want to fine-tune the model, we can analyze the performance of the model under different noise types, or MTO and hippocampus subregions, or on the whole brain. We could also investigate the correlation between longitudinal MRI change and molecular hallmarks or cognitive changes. And that's it for my presentation. And I would thank you and thanks my previous lab for supporting me and being here to listen to this. Thank you. Yeah, if you have any question, please go ahead and ask.
Yeah. Thanks a lot, Mengjin. That was very nice and informative. I have a question. I was wondering, how could this framework be applied for large clinical studies? And one more question is, do you notice any are there any noise sources in the longitudinal MRI scans that you use? And if so, how do you take care of those?
I think our algorithm can be useful for clinical studies, because we have shown that the algorithm will require a smaller sample size, so it will be good for, and also it's good for detecting subtle changes at an early stage. So it'll be good for clinical studies to cut their budgets by recruiting just less people. And also, because in Alzheimer's disease, there are some other comorbidities, I guess I can also use this algorithm to analyze different, like the different performance of comorbidities to see the difference in progression. And as for the second question that you asked about noise, actually, I had a backup slide for different sources of noises that I summarized later on. And basically we can categorize those noise sources into biological and non-biological, and some of them are systematic and some others are random. So we only want to detect this biological difference with neurodegeneration, but for other sources of noises, such as scanner change or MRI artifacts, we would like to ignore them. And the way for our algorithm to control that is that we, how to put it, like for this column of non-systematic noises, we are controlling it by just extracting time relevant, right? These are time relevant, and these are not relevant to time. So I think the algorithm can, by nature, can ignore these ones. And then when we are controlling these like scanner changes and chronic ARIA, I think we just control our data set to be the same scanner group. For now, we don't mix different scanner upgrades. And for treatment-induced algorithms, currently in ADNI data set, we don't mix people who are undergoing treatment. But in clinical trials, because people are all going through that, I think we probably need some more effort to make sure to distinguish between ARIA versus others. We have other scanning techniques to tell whether a person has ARIA or not. So I think it should not be an issue either. Yeah.
Yeah, well, thanks for the detailed answer.
Thank you for the great question.
Does anyone else have any questions for Mengjin? You can also reach her out at her email if that's fine. Yeah, I think there's a question in the chat. Mengjin, if you want to read and answer.
Okay. Yeah. Thank you for the presentation. Do we have any benchmarking data set or ground truths like corresponding noise-free images of a participant image? And without corresponding noise-free ground labels, how can we use synthesize in models like GANs? Yeah, I think that this is a great question, and this is exactly the reason that we abandoned using GAN models for our analysis, because our hypothesis is that even with the most advanced MRI machines, there can be artifacts, or the image resolution is not high enough for a noise-free image. And because the hippocampus is really small, that has made the error more significant compared to the hippocampus volume. So we really want like very highly detailed images, but we don't have it for now. So that's why we didn't use GAN models for our analysis. Does it answer your question? Thank you.
I think if there are no other questions, I would like to thank Mengjin again, and also thanks everyone for attending on the weekend. It was a really great presentation, Mengjin. Thanks.
Thanks, everyone.
Thank you very much, Dr. Mengjin, and thank you, Pulkit, for organizing.
Yeah, thank you. Good.